So next we're gonna talk about how do they move? How do the substances move? So firstly, you need to know what are their molecular size, what are their poles, and what is their ionic charge. So for the substances that can dissolve in lipids, they must be non-polar. For example, fatty acids, glycerol, fat-soluble vitamins A, D, and K, and steroid compounds. While lipid-insoluble substances consist of large molecules and also small molecules that are polar, some non-polar molecules like oxygen and carbon dioxide, and ions. Again, remember what I said about the experiments. So, passive transport is the main thing about passive transport is that it doesn't require energy, while active transport requires the use of energy. So the three types of passive transport that you normally come across are simple diffusion, facilitated diffusion, and osmosis. So simple diffusion is moving molecules from a high concentration to a low concentration, and this occurs like anywhere. And a main example is like oxygen. Oxygen, they don't dissolve in lipids, but they can bypass the phospholipid bilayer, and they only tend to bypass the bilayer when there is an uneven, when there is no dynamic equilibrium yet. What that means is basically when one region has more oxygen than the other, then more concentration of oxygen than the other, then it will pass through the phospholipid bilayer. For osmosis, it is like diffusion, but instead of the molecules moving from one place to another, it is the water that moves from one place to another. So basically, the best way to say this is, when one solution has a higher concentration of certain stuff like glucose or sucrose than the other, it has a low water potential. So so what that means is, you can say that it has not enough water, I guess you can say. When the concentration is high, if the concentration of the solute is high, it has a low water potential. So what that means is it doesn't have enough water. So and vice versa lah. If there is a low concentration and it has high water potential. So again, we say it moves from high to low. So water moves from the lower concentration to the higher concentration. So instead of the molecules moving from a concentrated region to a less concentrated region, it is the water that moves from a less concentrated region to a more concentrated region, so that they both end up having the same concentration, which is dynamic equilibrium. Next is facilitated diffusion, and if I'm not wrong, it still has movement from a higher to a lower concentration. Remember, simple diffusion, passive transport is from a high to low, and all of these do not require energy. But facilitated diffusion needs these carrier and channel proteins to help transfer these nutrients. So how does it work? For channel proteins, they're basically special like tubes for certain ions or molecules, and they have like these. It's basically like a security system where only some can pass through and some cannot. While carrier proteins, they are specific. And like for example, for channel, they might allow certain ions, like more than one ion, but carrier proteins only allow glucose, for example. And then like this side only allows glucose, this side only allows something else. So what happens is, at first the bottom part closes. Okay, if the concentration of the outside is higher than the inside, the bottom part closes. It lets the glucose in this case it's glucose, and then after it goes in, the upper part closes and the lower part opens, so the glucose can transfer into the cell, and this repeats itself. Active transport requires energy. That's the main thing about energy, and the energy is from ATP. And remember, ATP comes from the mitochondria. So again, I said it moves from low to high because it is the opposite direction. It has to go against the flow, so it is low to high, and it requires carrier proteins. For passive transport, it doesn't need proteins, or if it's for facilitated, it requires channel or carrier proteins. But for active transport, it is only carrier proteins. So what happens is when ATP molecules attach onto this like attachment site, it will so-called release energy, and that's how the thing opens and closes. So here's the most popular example. It is the sodium potassium pump. So firstly, these 3 sodium ions, it says 3 in particular, that's just how the carrier protein is. They bind to the carrier protein, and then the ATP will decompose into a phosphate ion and ADP. So the phosphate group will bind to the carrier protein. So basically it goes to the attachment site, and the attachment site, and when it does bind to the attachment site, it causes energy to go into it releases energy into the carrier protein. So the carrier protein will change shape. It will open the outside and close the inside, so the 3Na can come out. And then after that, the potassium ions from the outside will come in instead. So at first the sodium goes outside, but this time the potassium goes inside. They take up the spots in the carrier protein. So there's 2, and then when the phosphate group leaves the attachment site, it releases energy again, and it releases energy again, but basically when it leaves the site, the shape changes back to normal, and that's how the potassium ions go in, and then the process repeats itself. Okay, it says here there's another type of pump known as the proton pump. So they don't normally talk about this much. If they do ask about this, they'll talk about the sodium potassium pump. So read this yourself. So yeah, I mentioned a lot of this already. Passive transport doesn't require energy. Active transport is from low to high, which is against the gradient. The way to write it as an answer will be it occurs against the concentration gradient, and then active transport will cause cells to either accumulate or dispose. Like if you use passive transport, both sides must be 50-50 in terms of concentration. But if active transport, you can completely empty out the site, and this one will have everything else. So yeah, but they both must require they both must require the substance to move through a membrane now. Movement of substances across the plasma membrane in living organisms. I'll continue this since it's related to the subtopic. So what is the main examples of passive and active transport? For passive transport, you will normally see this in gaseous exchange between an alveolus and a blood capillary. So this is simple diffusion. You'll see this more in the examples in like the next upcoming chapters. And then osmosis occurs in the kidney to reabsorb water. Again, you'll learn this more in another chapter. Absorption of a water by a plant root vessel by osmosis. This one is quite popular in exams, but again, another chapter. And Absorption of fructose molecule in the villus by facilitated diffusion. This one isn't that common. Next, active transport is the absorption of glucose and amino acids in the villus. Next is reabsorption of glucose through in the kidney again. Transport of sucrose from a leaf to a phloem, and absorption of mineral salts by plant root hair cells. Again, these all get repeated in the next chapters. So yeah. Now this one. This thing keeps appearing in the forecast papers that I was doing. These solutions: isotonic, hypertonic, and hypertonic. So isotonic means same concentration. Hypotonic means low solute concentration, and hypertonic means high solute concentration. So what that means is, if one solution, remember this is between two solutions. For example, maybe a white blood cell and water, sucrose solution. So if one solution is more concentrated than the other, we say that it is hypertonic to the other solution. If the solution is hypotonic, then it has a lower concentration than the other. So when A is hypotonic to B, water will go from low to high, right? So water will leave A and go to B. For hypertonic, A is more concentrated than B. So that means that A is hypertonic to B, and water will move from B to A. And it moves through osmosis. Remember this is very important. So you see this a lot often. I mean you see this question very often in objective. So what will happen when these red blood cells are placed in hypertonic, hypotonic, and isotonic solutions? In isotonic solutions, water will go in and out through osmosis at the same rate. So that means that the shape does not change. But if it's in hypotonic, remember, hypotonic means hypotonic, when we talk about hypotonic, we mean the solution, not the red blood cell. The red blood cell is hypertonic, but the solution is hypotonic to the red blood cell. This means that the hypotonic, the solution has less concentration. So water will move from the less concentrated region, which is the water, the solution, into the red blood cells. Since water goes into the red blood cells, it causes the cells to overflow. Basically, it cannot tahan anymore. And when it cannot tahan, it will burst. When it bursts, it's called hemolysis. So why does it burst? Because the plasma membrane cannot tahan anymore. Why cannot it tahan anymore? Because it's just not durable enough. Because the plasma membrane is too thin. Next is hypertonic solution. When red blood cells are in a hypertonic solution, which means that the outside is more concentrated than the red blood cell, water will leave the red blood cell and go to the more concentrated solution. Since water leaves the cell, basically there's less water in the red blood cell, so the cell will shrink. And this is called crenation. For plants, when you have a hypotonic solution, which means water will move into the plant cells, it will cause the cells to become turgid. It will cause the cells to become turgid. So unlike animal cells, plant cells do not burst because they have cell walls, and cell walls are meant to maintain shape and provide support, right? Remember? So yeah, this is very important because if your cells are all flaccid, which means they are not turgid. Remember, when they are packed, we call it turgidity. When they are full, we can say it. If they are not turgid, the the stem of the plant would be very brittle. It will easily break, it might wilt. Okay, and then next will be hypertonic solution. So this is when water will move out instead. And when water moves out, the vacuole and the cytoplasm will shrink. The cell wall is fixed, so it doesn't work, so it doesn't like move as well. But the vacuole and cytoplasm will shrink, so some of them will be detached. Not the whole thing will not be detached, because then the cell will die, but part of the cell will detach. So it's something like this. If this is the cell wall, your cell will look like this. So some of it is still attached here, but it is flaccid. Flaccid means it is basically not full. It is almost empty. So this is called plasmolysis. And as I said, if it is not turgid, the cells will become brittle. I mean, the stem will become brittle, and the plant might wilt. But however, unlike crenation, if you can put water back into the plant cells if you put it in a hypertonic solution, and when you do that, it becomes turgid again. So that's called deplasmolysis. So for isotonic solution, they again nothing changes, because it's not completely turgid. We call it, it's still flaccid for us. Yeah, okay, that's all experiments. Okay, these are the applications. I think I'll just bring this up. So number one, plant wilting. We already mentioned this. When plasmolysis occurs due to water moving out of the cells through plasmolysis, through osmosis, the plant will wilt and eventually die, because remember, they don't have water. Next, rehydration drinks. I haven't really seen this much. Isotonic drinks. I don't like isotonic drinks. Saline solutions for blood plasma, and liposomes. So yeah, these are all applications. So these are all up to you to read.